In today's episode, we speak with Dr. Arnau Sebé-Pedrós from the Center for Genomic Regulation in Barcelona, whose research bridges evolutionary genomics, single-cell transcriptomics, and developmental biology. By studying early branching animals and their unicellular relatives, Dr. Sebe-Pedros reconstructs the emergence of cell types, gene regulatory networks, and multicellularity. What can the cell atlases of sponges and plastozoans reveal about the ancestral toolkit of animal development? How did transcriptional and epigenetic programs evolve to drive cellular differentiation? Join us as we trace the deep roots of metazoan complexity.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
So today I'm joined by Arnau Sebé-Pedrós from Catalonia, I guess that's how it's pronounced. Welcome, Arnau, on the show.
Yeah, thank you, Stefan, and thank you for the invitation.
So a question I like to ask every guest to start off our little podcast and to ease into the topic is, how did you become interested in biology in the first place and then in pursuing a career in science?
Okay, so I grew up in the countryside in Catalonia, northeast of Spain. So I was surrounded by farm fields, and so there was not much to do, to be honest. So as a kid, there's no scientists or academics in my family, but we used to go out a lot to nature. We spent our summers in the Pyrenees, hiking and camping. So I got kind of like naturally interested into first it's more like insects, then birds, and so I was always keen on nature. Even as a teenager, I started working then a bit more on with groups that they were doing like radio tracking of stepland birds and doing censusing of insects and micro mammals that they feed on and so on. So I was kind of like slowly getting more and more into let's say more like field work and biology. So it was a very obvious choice for me to study biology. I was initially more interested in ecology, zoology, and so on. But then I discovered evolutionary biology. I got completely hooked with it, and also more like molecular biology, genetics, biochemistry. So, and that was my path into science. So I always thought I would be a field biologist, but I ended up more into molecular and now also computational.
Yeah, I'm talking about that. For those who don't know you, could you introduce yourself and your current role?
Yeah, so I'm a junior group leader at the Center for Genomic Regulation in Barcelona.
So you are, no, I can't see it, but I guess from from your lab, you can see the ocean, right? So that's...
Yes, almost. I mean, in one direction, yes, we're right in front of the sea.
Yeah.
Yes.
So if I may quote your website, in your lab, you apply and develop single-cell transcriptomics methods to unbiasedly characterize cell types in whole adult organisms of diverse animal lineages. We focus our single-cell sampling efforts mainly on non-bilaterian lineages. Porifera, Ctenophora, so this will be a hard episode for me to pronounce all those names. Placozoa and cnidaria. Since on this podcast we mainly focus on mammalian model organisms and maybe sometimes on plants or nematodes, could you give us a brief intro into the model systems you are using and why they are so useful?
Yes. So basically we kind of everything we do in the lab, so some work on single cell, also a lot of chromatin biology. The kind of a guiding principle is comparative biology. We're interested in evolution and we approach it from a comparative perspective, which means that the guiding principle that determines our selection of species to study is always a phylogenetic tree. And in that sense, for some of the questions at hand, which is the evolutionary origin of animal genome regulation and cell type diversity and kind of the regulatory logic of cell type specification, the useful set of species to look at are these non-bilaterian species. These are the earliest branching animals in the tree. So if you want like our more distant cousins in the animal tree of life. So then we can compare them with us, with the other model species, and also with outgroups. It's also very important for us to. That's what I did my PhD on, the unicellular relative of animals as well. So this is for this kind of the main topic in the lab, but we also work a lot on diverse eukaryotes for questions about chromatin evolution. So that's a bit like what is the guiding principle. Many of them are as far as it gets from a model species. And that's one of the beauties of these single cell and chromatin technologies, that we can get less and less dependent on our capacity to even keep these animals alive in the lab and sample them directly from the field.
So let's start with a paper that came out in 2018. There you performed whole organism single-cell transcriptomics to map adult and larval cell types in the cnidarian Nematostella vectensis. Could you talk us through how you got into this and what you found there?
Yes. So the way... So in my postdoc, single cell was never the original plan. Actually, it was plan B at the time when I joined the lab, Amos Tanai's lab at Weizmann. The main focus was to be 3D chromatin, but things were not working very well. So we turned into plan B, which at the time, 2015, the lab of Amos and Idoami, they developed Marsec. So I started playing with the technology. And Nematostella was one of the obvious species to look at because it's probably the closest thing, together with Hydra, to a model species in Cnidaria. So this is a model species developed by the group of Ulitech now in Vienna, also Mark Martindale now in Florida. So we started collaborating also a bit by accident with the group of Francois Spitz and Heather Marlowe in Pasteur, because they were collaborating on other things with the group of Amos. And so we started with Nematostella. The beginning was not working very well, but then kind of after a few rounds of tests and so on, things started to work. So it was an obvious place to start, as it's a system where some things are known, and also where we could go back to the organism and do in situs, even transgenic reporters to validate things. So it was one of the first published whole organism cell atlases for a non-model species, and for us it was a fantastic training on where we could go with this data, how to analyze this data. It was really transformative, even from this from a personal or from a scientific perspective, on what is the potential, which is obvious. I mean, of course, everybody saw that for non-model species, evo studies, single cell technologies would be transformative, because basically we can now go and deconvolve the whole cellular complexity in organisms where we cannot generate stable transgenic lines, enrich or cut tissues and organs and so on. So this kind of opens up the unbiased characterization of cell types across species. So that's how we got to work on Nematostella. Yeah.
Sorry to interrupt you. So did you have to do like pioneering work, how to get single cell studies to work in those Nematostella? Or has there been studies that already did that?
No. In that sense, single cell work, there was nothing. I mean, nothing even in anything that was not at the time. Even the first C. elegans paper from Jason Liu was not published when we started working on Nematostellas. It was mostly human mouse, a little bit of Drosophila data. So we didn't. It was. I should say also another important criteria, that this is something we may talk later as well, that has changed dramatically in 10 years, is that choosing Nematostella among all cnidarians was an obvious choice because of the constraint imposed by the availability of a genome sequence. For a long time, for all these our favorite lineages, sponges, the Ctenophores, cnidarians, there was one genome. Many of them sequenced by the Joint Genome Institute in an early effort in the late 2000s. So that was it. Like, that was the obvious species of choice if you were working on, in this case, cnidarians. No?
How easy
it was very important.
Yeah. How easy is it to grow those animals?
It's easy. Nematostella, it's quite easy to grow. I mean, we still grow it in the lab, and it's quite low tech. So it's quite easy. So yes, we started with the easy stuff, in the sense that we could culture them. The genome was relatively okay. And basically it was about cracking dissociation and also some tweaks in the molecular biology, and then mostly also on how to analyze this data. At the beginning, we were a bit I would say, underwhelmed by the what we thought was some intrinsic problem with the sensitivity of the method. The number of transcripts we were recovering per cell was relatively low compared with what people are used to or were used to in more like in cell cultures and so on. Over the years, we've learned that it doesn't get much better than that. So there is an intrinsic property, which is species-specific, cell type-specific, also age-specific into the transcriptional output of a cell, and we need to live with that. Okay. That was actually one of the strong motivations at the time that in Arnau's group, they were developing the Nematostella algorithm, because it was a good way to integrate this very, very, very sparse data into something that we could work on.
Okay.
And do the kind of things that people would typically do with single cells, with these aggregate metacells.
So next, you used whole organism single-cell RNA sequencing to map cell-type-specific transcription in Porphyria, so sponges, Stenophora, comb jellies, and Plazozoa species. I'm sorry for that. So yes. What did you find there in comparing those?
Yeah, so then, so once this worked in the Nematostella, we kind of quickly moved into looking at other species, again, guided by the fact that these were the quasi-model species with the genomes available. I knew from my past work during my PhD, some of the people working on this. So we had these sometimes 1 week long jamborees of people coming to the lab, sometimes from far away. Like I remember Federico came from Australia with sponges from Bernie Degnan's lab. I picked him at the airport with a taxi. We went to the lab and we spent the whole night in the fax, whole night sorting plates, Marsec plates for the sponge atlas. So that was it. It was one night of sorting, because then the next day the cells were not, the sponges, were not so happy anymore. So now we do things very differently. We fix, so we have become much more detached and independent from the sampling to the single cell profiling. But so it was a bit kind of like the logic steps of looking into the known. In retrospect, it was not the easiest also to interpret those atlases, and even from a comparative perspective, starting by comparing a sponge, a Stenophora, and a cnidarian, it's a bit tough. So there was an element of novelty, but then, for example, we also had many doubts about the annotation of certain cell clusters, and even from a comparative perspective, comparing cell types in a sponge and a cnidarian, it's not the most straightforward.
Can you draw any conclusions from those comparisons?
Yeah, we could draw. So, and this is actually something that is a particular research line that we are very interested in the lab, which is we could start. So for us, it's a very useful tool to catalog cell types and then to follow up on the biology of the cells and so on. But for us, these whole organism atlas, it gives us an idea of the different readouts of the genome. And in that sense, going back to the genome, we added some chromatin profiling, we did some ChIP-seq, some attacks, and so on, and starting to try to decode the regulatory information, determining the cell state and how this differs between, let's say, early branching animals with very few cell types, and animals with more cell types. At least from a data-driven perspective, we can talk about animals with more cell states than others. This was interesting. For example, the fact that simple placozoans do use predominantly promoter-based regulation with not so fancy distal cancer elements and so on. This actually motivated some of the follow-up research that we've been doing in trying to explore these regulatory landscapes and so on. So, but that said, it was very initial exploratory work, descriptive work.
Okay. So to get started and set the ground for other work, yeah?
Yes.
Then you looked at the cell types and molecular pathways that underpin the biology of reef-building corals by using single-cell RNA-seq. So were they also brought from Australia?
No, this was one of these. I mean, this was one of the things that happened just because I was in this very stimulating scientific environment, which was the Weizmann Institute and Israel in general. And we started, I don't even remember how, we started this collaboration where we met with the group of Talimas and with Shani Levy, who later joined my lab for a postdoc, that they were working in corals in Haifa. So these were Red Sea corals. So this was our next attempt to try to work on species that we could not culture in the lab. So we took them from the wild, again with some intense sessions of sample processing. So it was interesting in two levels. One, to attempt to work with, let's say, non-model species, things that we could not even grow in the lab. And second, it allowed us to get into more comparative work. So now it made sense to compare cell types within cnidarians. So at the time there was Hydra, was also from Selina Juliano, published the Hydra Atlas. So this was kind of the first work from my independent group. This was nice. Like, it makes sense to start comparing things that are maybe a bit closer, to try to see things that resemble each other and how evolutionary conservation fades away with time. So this was interesting both at these two levels. Three levels, I should say. One is the biology of corals, which is very interesting and fascinating on its own. From a comparative perspective, we could compare 4 cnidarian atlases. And also from a methodological perspective, with taking these further steps into sampling as many species as possible outside the lab.
Yeah. And this maybe paved the way to the next study where you reconstructed eukaryotic chromatin evolution by comparative proteomics and genomics analysis of chromatin eukaryotes and archaea. Can you talk about this approach and what you found there?
Yeah, this is kind of like the secondary research line in my group, and something that I'm always been very interested and keen to continue. So this has nothing to do with the single cell work, but more about understanding the mechanism of genome regulation.
Yeah.
And in this case, the relevant phylogenetic context is eukaryotic evolution. Chromatin is an eukaryotic novelty. And here what we did is we selected a number of species, protists, mostly some of which I already started working during my PhD, that we can culture in the lab. Again, the species sampling decision is based on availability of cultures in this case, and also availability of genomes, which is still relatively limited. So here we started focusing on describing histone modifications. Now we did some follow-up work, so we know what are the building blocks, histone marks. Now we profile them with ChIP-seq in different species to see that these histone marks define different chromatin states in different species, some of them, the repressive ones. And we are following up these also now and trying to study the evolution of the readout. So what are the readers and complexes that built around these very conserved building blocks, which are histone modifications across eukaryotes. So it's a bit different topic, but it's connected. In the common theme is the evolution of genome regulation.
So do you see that along the evolution, there are more and more histone modifications joining the pool of chromatin, or is it like all of them were present in the beginning?
No, not at all. I mean, it looks like, of course, we have a certain ascertainment bias. I mean, even though we use mass spectrometry to focus on the conserved ones, there is a lot of novelty on histone variants and even their own modifications. But there seems to be a very conserved set of marks very much present in most, if not all, species. And even their readout seems to be quite stable, especially activation marks and so on. So at least thinking of then the specific context of an increase in complexity of genome regulation, which is what we thought happened in the transition to multicellularity, it was not based on more building blocks. I think using these things somehow in a different way.
Yeah, very interesting that once chromatin is established, it's like the version 1.0 is like already the full set. It's feature complete.
Yeah, something must have happened. There are hypotheses, on the invasion of parasitic genomic elements and so on that determine, it's quite remarkable even that post-translational modifications are so conserved across billions of years of evolution. Yeah. Then the repressive chromatin, something else happened there, probably because there is an ongoing kind of tug of war between these parasitic elements and the repressive marks. And this is how things may have changed. So there we see losses, we see repurposing of these functions and so on. At least the process of activation of a promoter with a series of specific histone marks, this seems to be quasi-universal thing in eukaryotes.
So one thing that is different along evolution is the neural system, right? And to analyze this in more detail, you reconstructed the process of the assembly of neuronal programs by investigating possible neural affinities using phylogenetics, chromatin profiling, and comparative single-cell genomics. What did you find there?
Yeah, so this is again another study where we decided to go a bit taxonomically deep in one of these lineages that we are interested in, Placozoans. And the initial motivation for this was not at all to try to understand anything about neural evolution, but more like we said, we thought that Placozoans would be a good model for cell type comparative biology, because we thought that there would be few cell types, a few species that we can culture, a few genomes, so that we could start to model, the evolutionary dynamics of cell type identity transcriptional programs with the ChIP-seq, with the ATAC-seq, and so on. To our surprise, we discovered, which some of them were known, but we discovered a much higher diversity than expected of these peptidergic cells in Placozoans. Basically, cells that share some of the neural gene expression program, primarily the neuropeptide processing machinery and the presynaptic scaffold, and that are used to control by paracrine signaling collective cell behaviors. In this is just for the audience to imagine. A placozoan is like a few millimeters flat 2-cell layers where probably paracrine signaling is effective enough to control these behaviors. When I say cell behaviors, I mean contraction, ciliary motility, mucus secretion, secretion of digestive enzymes, and so on. So there's a series of collective behaviors that seem to be controlled by these cells. So it was known from the genome analysis that placozoans encoded for some predicted neuropeptide proteins, neuropeptide or secreted peptides that even were experimentally validated, some of them. But what was a surprise was to see so many different cell types producing different combinations of these peptides. And of course, this allowed us then, because we had the atlases of the ctenophore, of the sponges, of cnidarians, and other species, to do a cross-file comparative analysis of these let's say, protoneuronal cell type transcriptome and see what elements were in common and what could be novelties in evolution. Again, still the resolution of this is relatively low. We don't have any species to do this reconstruction, but it's an exciting first step that builds on these atlases that have been accumulating over the years.
Can you comment on the elements that were shared and which were new?
Yes. So the main thing, if we assume, which I think it's pretty reasonable, the cnidarian neurons and bilateral neurons, or closest relatives, are homologous. In the main, so the shared thing is all the neuropeptide processing machinery, a lot of GPCR signaling, which are the most obvious receptors for these secreted peptides, and the presynaptic scaffold. What was novel was electrical signals, tons of ion channels, which is one of the hallmarks when we look at a new atlas and we look for the neurons. We count GPCRs and ion channels, and a specialized postsynaptic scaffold to receive these signals. Those were two clear novelties of the cnidarian bilaterian neurons.
So you said that you used ATAC-seq and ChIP-seq to study those things. How was it to implement those methods in those model organisms? Did you have to do a lot of optimizations?
Not really. To be honest, I mean, ChIP-seq is pretty, at least in animals, pretty robust. It works well every time we try it. In practice, it's a bit more annoying because of the chromatin preps and so on. Can get quite annoying. But in animals, in general, it works quite well in our hands. Usually, an issue is finding the right antibodies. So this is something we worry about, and we try to put some effort. I mean, knowing at least from mass spectrometry that a mark is there or should be there or not, and then trying to be a bit paranoid about unspecificity and so on. And this can be tricky sometimes, especially for repressive marks, 27-trimethyl, K9-trimethyl. This took us some effort. But no, I would say in general, no. And ATAC-seq in general also works. It's quite straightforward.
I guess the most important thing with ATAC is the genome size, right? So that you get the amount of enzyme, right?
Yes, but in a way you can play it safe and titrate a little bit, and you will get one library right. So I say generally in our hands it has worked well. Whole different story with single cell attack. Single cell attack has been quite a nightmare for a few years for different reasons, which is more like nuclei prep and so on. But attack-seq is relatively straightforward, except in one group of organisms, which are ctenophores, that we've been trying, I always say I've been trying for 10 years to do an attack-seq in ctenophores, and we once semi-succeeded, and we don't know exactly why is that. I mean, I have really no explanation. Everything is weird with these organisms. Ctenophores, people may know them because they have this jellyfish-looking organisms that maybe they invented the nervous system independently from the rest of animals, and everything is quite special with their biology. And definitely there is also something with their chromatin that we still don't understand.
Okay.
And attack-seq, it's notoriously difficult in ctenophores.
I see. Okay. So to trace the evolutionary origins of animal genome regulation, you characterize the physical organization of the genome in non-bilaterian animals, so sponges, ctenophores, placozoans, and cnidarians, and their closest unicellular relatives, by combining high-resolution chromatin conformation capture with epigenomic marks and gene expression data. So what did this reveal? I guess when you say, or when I say chromosome conformation capture, this means 3C or high C, probably?
Yeah, micro C in this
Micro
case.
C, yeah. Okay.
So this is something that that's what. So the idea to look at this that was my original postdoc plan, and there were, in short, two limitations at the time. One was the quality of the genomes. The genomes were scrambled and very partial and problematic to interpret the data. And the other was the resolution of high C. At the time we were testing 6 nucleotide cutoffs, 4 nucleotide cutoffs. At some point we realized that, we had to go all the way back and sequence the genomes of the species we wanted to look at. That was a bit tricky. The motivation for this, I think the context is, and it relates to my work of mine and others in my PhD, where we were studying, people were doing comparative genomics of the transition to animal multicellularity of the origin of animals. At the end of the day, foundational work from Nicole King's group in Berkeley, then Iñaki Ruiz-Trillo, where I did my PhD here in Barcelona, was that gene innovation was maybe not such a big thing to explain animal origins. And then maybe one of the possibilities we should turn into genome regulation and how these genes are used together in different contexts, implementing epigenetic memory and this and that. And there was a very nice short opinion piece from Giacomo Cavalli and Amos Tanai back in the day that I read, and I was very excited about maybe the role of 3D chromatin-based regulation on this transition. So this seemed kind of an obvious next step to some of the work we were already doing at the time with trying to hunt for enhancer elements in unicellular relative of animals. So fast forward 5 or 6 years, when I started the lab, I decided, okay, let's take these two steps back. We started from scratch. We resequenced the genome from scratch of all the species we wanted to work on, which was a great idea. And also we started testing higher resolution, high-C methods. So we tried, the general interstitial enzymes, DNA, high-C, and then we tried micro-C, and we clearly saw, this is it. With this, we can really get there. So Diana, very talented postdoc in the lab, started generating data on again, by now, this classic species that we always work on, our favorites, to reveal that we had some hints from ChIP-seq and from attack-seq and so on, that there are distal elements, even in some cases pervasive loops contacting or linking these distal elements with promoters, at least in some of these early animals, which was quite exciting and quite nice to see. Now, of course, this opens many questions about the mechanisms and whether the mechanisms are homologous or not. But as a first step, this was,
But you see basically the same TUT-like genome structure as with mammals?
Yeah, so that's interesting. So we try to avoid that concept in the sense that sensu stricto, and the way we try to frame it from a comparative perspective, is that we can say that they are sequence-driven, so elements in the genome with specific sequences, which even in one case we managed to chase with proteomics the factor, C2 losing finger that seems to be binding there, that there are some sequence elements that determine the formation of these structures, in this case loops. Now, if we apply the same concept to insulation, an active insulation that is not explained by passive segregation of chromatin states or transcriptional status of a promoter, this we couldn't find. So what we propose is that, yes, loops we see in the earliest branching animals, whether they are produced by homologous mechanisms or not, that's the next question. So, loop extrusion, the same factors determining these sites, and insulator elements in this species we haven't managed to identify. Again,
so there is no
with
CTCF?
Conclusions. No, there is no CTCF. I should have started with that. No, there is no CTCF outside bilaterians. But in this case, we found a C2H2 zinc finger that is methyl sensitive. We could validate that as well with DAPSIC. That it's there in these loops in ctenophores, and it's a ctenophore specific protein. In the same way that there are many insect and arthropod specific proteins involved in the Drosophila chromatin architecture, right? BIF32, CP190, and so on. So assuming that cohesin has something to do with this, which we saw cohesin enriching the sites using ChIP-seq, SMC1 ChIP-seq, but we couldn't find any stripes or flames, anything suggestive of some active loop extrusion. Could be that because we are working with adult organisms like this is, so now we are looking at some developmental stages as well. Or it could be that this is not involving loop extrusion and there is some just a game of stabilization of this contacts. For context, the contacts that we see in this species are between 20, 30 kbs away.
Mm-hmm.
Okay, so promoter enhancer contacts. So that's why we needed resolution. So that's one thing. Interestingly, in cnidarians in the Nematostella, we see way less loops, only in some of the classic transcript genes, transcription factors, and so on, and they show one-sided flames from the promoters, which may suggest that there is some specific loop extrusion activity from there. Again, that's something that we are following up on.
So you just indicated the distance. So in general, can you say that the distance of the regulatory elements which interact is smaller than in higher eukaryotes?
Well, definitely more distant than invertebrates, but I think this is largely a passive byproduct of the history of the genome, which is determined by, again, everything is a population history, no? So basically, dynamics of expansion of the genome by transposable elements, and that's the next thing also that it's obvious to do, is to compare chromatin architecture between species that have very different genome sizes within cnidaria. No? So could be that how pervasive or not are these is distal regulation. The loops, the formation to enable distal regulation, if we assume that this is causally linked, may be determined by some intrinsic property of the genome being larger or smaller. So if you have a more compact, streamlined genome, you may have less regulatory landscapes where you need some kind of stabilization of this. While if your genome has been expanded by whatever reason, possible element invasions, whatever, then we may see more of those. Interestingly, and this is connected with some recent experimental work from a group of Fish Nora and others as well, showing the distance dependency on loop extrusion, right? So this is just a correlate, but it's interesting that in sponges, in the one sponge that we looked at, we don't see loops. But turns out that it has a genome that it's relatively compact, but also all the potential regulatory elements that we identify by ChIP-seq and ATAC-seq, they are very close to transcription start sites or the nearest transcription start site. So we said, well, maybe it's this. It's not like sponges have lost the chromatin loops or they don't have, but maybe if we look at all sponges with larger genomes, we will see that this is there. Again, the only way to really trace the evolution of this thing is by narrowing down the proteins involved in, because then we can link them by orthology. microspecies. We can talk about a phenomenon that is the same, which is the formation of loops, but we need to know what are the proteins involved in the mechanism, which is a bit trickier.
Yeah. So a few episodes ago, and I'm just checking, it was episode 154. I talked to Sarah Teigman, and you also said that you listened to this episode, which is great. And we talked about the Human Cell Atlas. We also talked about other consortia on this podcast already. And to be honest, the Biodiversity Cell Atlas was completely new to me. Could you put us in the picture of the mission of this project consortium?
Sure. Oh, yes. Yeah. So basically, this was an idea. So we basically got together in 2023 here in Barcelona, funded by the Wellcome Trust. A number of people, so people from the Human Cell Atlas, actually people from Biodiversity Genomics, Earth Biogenome Project, Darwin Tree of Life, and so on. And then a couple of people that we were kind of in a more or less disorganized way trying to do single cell in non-model species. And we got together to say, well, maybe there is now, for the first time, the opportunity to start thinking about scaling up and systematizing cell atlasing across the tree of life. First of all, because now we are no longer restricted by one genome per phylum. I no longer need to work on Nematostella or Hydra if I work on cnidarians. There are many and super high quality genomes, chromosome scale genomes produced by the dozens, literally, in Darwin Tree of Life, for example. So this is there. And then maybe with a concerted effort of learning of all our failures and frustrations over these years in why, sometimes single cell it's a bit different. So transcriptomics in this case, we can accelerate this, and then also to think about, but then why would we want to do this from different perspectives? People that are interested in genome biology, in organismal evolution, from different perspectives. So we had the second meeting in Woods Hall last year. Again, other people, or some of the same people, and we put together a white paper, which will be out next month, hopefully, laying out what are the potential and limitations. And we even got some funding from the Moore Foundation to start what we call phase 0, which is basically trying to systematize a little bit sample prep and single cell methods. So try to see how to streamline and come up with these species of lineage validated protocols in protocols.io, and also we are building a database to host this data to allow for species comparisons. And the idea is that with these experiences to then move on to scale up atlases, initially with transcription, single cell transcriptomics, but for example, my group and others involved in this were particularly interested more on the chromatin, like chromatin accessibility and looking into the seed regulatory codes. Others are more interested in the spatial dimension. So I think, but there are many things in common, both on the data analysis level and in the experimental problem-solving levels that can be shared. And the idea of creating a community where we start sharing these know-hows and do's and don'ts, and we always say we will not have a magic recipe for producing an atlas in any new species that you might collect from the sea. But we can come up with a sampling decision tree where it says, okay, try A. These are the QCs that will tell you if A is working or not. Go to B. Consider doing these or starting from these protocols that are out there to accelerate this. So again, the idea is to accelerate these, not only in my case, it's because I'm interested in evolution and evolution of genome regulation, but there are other dimensions to it. For example, the study of symbiosis. That's quite interesting. Cells within cells and what they are doing together, or viruses. I think there are beautiful examples of there, or studying giant virus infection dynamics in the ocean. So just to connect all of these, again, around the tree of life with the genomes and with the technologies and future, and also stimulate technology development, where we realize that we diagnose that there are bottlenecks to put some focused efforts on that. So, so hence the idea of biodiversity.
So it's still in the early phases, right?
Yeah, super early phases, yes.
Okay. So there currently is also a paper on bioRxiv called Diversity and Evolution of Chromatin Regulatory States Across Eukaryotes. Do you want to talk about this work?
Yeah, I mean, I kind of already alluded to it. It's more like, okay, we saw these stone marks here and there, but then we put some effort in, again, in a very small number of species, but in a couple of species that represent highly diverse, but completely unsampled, in some cases, chaotic lineages to try to see what are the chromatin states or what is the distribution in the genome of these stone marks. So we tested antibodies, we kind of optimized a multiplex chip method so that we could do many chips at once, but also test, kind of have internal controls on the chips by mixing species together with chromatin barcoding to try to slowly expand this understanding. So there are two dimensions to it. One is understanding what are these chromatin states, and again, this is where we see that the active chromatin states are super conserved, but that there is that repressive states change a bit in composition and also in associations with transposable elements, gene repression, and so on. But I think in the future there is an important dimension to this, which is okay. We're producing all this genome. Earth Biogenome Project, there are big consortia producing genomes, but a very tough. So sequencing and assembling these genomes has become pretty routine, and it's amazing the quality of these genomes. But then you need to annotate these genomes. You need to build gene models, which is challenging, and also to try to functionally annotate these genomes. And I think decorating these genomes with chromatin data would be probably the most straightforward way of providing this kind of functional annotation to these genomes. So,
Okay.
no.
Do you already have a timeline for this paper?
For the preprint?
Yeah,
no, it's currently under revision.
Okay.
So
let's see. So imagine you are due to submitting a grant proposal tomorrow. What would you have written into this?
Good question. So biodiversity cell atlas aside, I mean, for my group, is it a high risk, high gain, or a conservative one?
It depends on you.
Okay. So no, I mean, something I think I'm quite excited about something that I think we are posed to follow up on this, which is to start exploring cell type identities. I mean, cell type identities across species, not from their expression profiles, not from the transcriptomes, but from the seed regulatory information, so DNA sequences. So basically doing cell attack, multiome, and so on, and also understanding something about the transcription factors involved and their binding specificities, and see if there is some kind of shared seed regulatory codes across species. We've seen some pretty spectacular proof of concept working in that direction from the group of Steinerts, also Henrik Kaisman in the vertebrate, so mammalian and bird pallium and some. So I think that's quite exciting, and it's moderately, I would say, high risk and definitely high gain. Yes.
Yeah, then fingers crossed for that. So for the last 45 minutes, we've been on a journey through a scientific career. Did we miss something important, or would you like to add something? No, I think we covered most of our main works. I think I'm, if I just to emphasize something, it's how exciting it is, this feeling of tapping into the unknown. Every time we build an atlas for a relatively unknown species or new representative of a lineage, there is an element of discovery, always finding something unexpected, that it's really, really, really exciting and motivating on itself. And yeah, I think this revolution of functional genomic technologies in the past 10 years has really opened up a whole new not a field, but access to certain questions and even classic questions in evolutionary biology in Evo that were completely inaccessible just 10 years ago. So it's still worth reminding ourselves that this is still a lot to be done and very, very, very exciting. So to finish, and this is my last question, what is your personal definition of epigenetics?
Oh my God. Okay, so we skip the transgenerational definitions. At least this is not a word that we use a lot in the lab in purpose, but so I guess the way we use, at least we can say our personal or lab definition, is basically any information not encoded in the DNA. And because we think of cell types and cell type evolution, and also the evolution of the very possibility of differentiating cell types, I think epigenetic mechanisms as a way of implementing memory and ontogenetic memory into cell identities. It's, well, at least our narrow definition of it. Yes. That's why we tend to talk more about gene regulation, genome regulation.
Yeah, of course, that's the more general term, I guess.
Yes.
So thank you, Arnau, for your time and for being on the show.
Yeah, thank you very much.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.